Okay, moving on to section 3, we're just going to kind of do a little bit of hormone overview and how hormones act. A lot of our hormones actually act through second messenger systems or by activating very specific genes. So, if we're talking about hormones, we know that hormones circulate systemically because they have access to our bloodstream. Where most of them can be found traveling, but only cells with the receptors for that hormone are affected. So there is target specificity. Target cells with the receptors for that specific hormone are the only cells that can be affected by the hormone to begin with. Once we have the hormone bind its specific target on specific target cells, hormones are going to alter the cellular activity, usually by either increasing or decreasing the rates of some sort of normal cellular process. So, some examples of how hormones can act once they bind to their target cells. Basically, a hormone produces one or more of these following changes. They can alter the plasma membrane permeability and or the membrane potential. How do they do that? By opening and closing different ion channels. Also, they can stimulate the synthesis of enzymes and other proteins. They can also go in and activate or deactivate enzymes. Once they bind, they can also induce some sort of secretory activity. And finally, they can stimulate cellular division or mitosis. So, when we're talking about the actions of hormones, again, we're going to divide this up based off of their chemical structure. So, is the hormone water soluble, so amino acid based, or is it lipid soluble and steroid based when we're talking about the structure? So, water soluble hormones, that's going to include all of those amino acid based hormones that we mentioned previously, with one exception, and that's the thyroid hormone. The thyroid hormone is a lipid soluble hormone, even though it is amino acid based. So, water soluble hormones or amino acid based hormones in general, they act on plasma membrane receptors. Why do they act on plasma membrane receptors? Because remember, they are not lipid soluble, they cannot diffuse across the plasma membrane. So, their receptor has to be on the external surface of a cell on the plasma membrane itself. Anytime we have a hormone that's binding to a receptor on the external surface on the plasma membrane, in order for it to get its effect to the inside of the cell, they're going to have to act through some sort of second messenger system. So all of these water-soluble or amino acid-based hormones are going to act via G protein second messenger system. Our lipid-soluble hormones are steroid-based hormones, again, are gonadal and adrenocortical hormones, and then our one exception that was amino acid based, which is our thyroid hormones. Because these are lipid soluble, they're all going to act on intracellular receptors. And when they act on these intracellular receptors, they're going to directly activate genes. And again, because they can diffuse directly across the plasma membrane, that's where their receptor is going to be found. So let's talk a little bit more about the second messenger system for our amino acid-based hormones. We have two main second messenger systems. One is cyclic AMP, also called CAMP, or the PIP2 calcium second messenger system. So you may have reviewed the cyclic AMP or CAMP second messenger system before. Basically, if we're talking about a second messenger system, we have two messengers. We have the first messenger and a second messenger. The first messenger is going to be the hormone. So the hormone binding to its receptor, that acts as the first messenger. Once that we have the hormone binding to the receptor on the plasma membrane, it's going to activate a G protein. And the receptor is going to change shape once it activates the G protein. And basically we have GTP involved, which is why it's called a G protein. If GTP is bound, then it's going to be on, so it's going to be activated. If we have GDP bound, then our G protein is going to be inactivated. So once we have our activated G protein, with GTP bound, our G protein can go on and activate or inhibit some sort of effector enzyme. Typically, the effector enzyme is adenylate cyclase. Once we have adenylate cyclase, then converts ATP into cyclic AMP or CAMP, and CAMP is actually our second messenger. So the first messengers are whatever hormones bound to the receptor, and our second messenger is going to be our cyclic AMP. And then once we have our second messenger that's formed, it can go on and activate protein kinases. Anytime you see any sort of kinase, that means it's an enzyme, and that enzyme specifically is going to phosphorylate something. So it'll phosphorylate some other proteins. Once we have phosphorylated proteins, those can go on to either become activated or inactivated. And we also have an enzyme in that area that's capable of going in and readily degrading cyclic AMP, and that enzyme is phosphodiesterase. Phosphodiesterase is really important so that you can stop the cascade that's occurring for the phosphorylation. So each activated adenylate cyclase means that we're going to have a higher number of cyclic AMP, and each kinase means that we're going to catalyze hundreds of reactions. So we have one hormone molecule that can actually end up with millions of final products because of this cascade amplification effect. So here's a good image kind of showing you what's going on. So our hormone's going to bind to the receptor. That's step one, and hormone is our first messenger. That remember, because we're talking about amino acid based hormones, the receptor has to be located on the surface of the plasma membrane, which is shown here. So once we have our hormone binding to its receptor, that's going to cause our G protein to become activated. Here's the G protein that is bound to the receptor. It's activated by the binding of GTP. So GTP means that it's activated, GDP means it's inactive. Once we have our G protein activated by having GTP bound, our GTP G protein is going to translocate across the plasma membrane until it binds to adenylate cyclase. Once we have our G protein GTP complex bound to adenylate cyclase, our adenylate cyclase is going to convert ATP into cyclic AMP. That is our second messenger. Once we have our cyclic AMP, that can go on to activate some sort of protein kinases, depending on which cell we're in is going to determine which specific kinase it is. But regardless, once we have a kinase that's actually functional, that means it's going to go on and phosphorylate something. So we have some sort of active protein kinase. But remember, just because we have an active protein kinase doesn't mean we're necessarily activating something. Sometimes we can go in and phosphorylate a protein to cause inactivation. So really the effects of our activated protein kinase are different. All of this is an amplification effect. Based off of one hormone, we can get millions of final products because of this cascade that we have going on. Okay, our second messenger system is PIP2 calcium. Some hormones do use second messengers other than cyclic AMP, and this would be one example. So we have our hormone activated G protein. It's going to activate a different effector enzyme. So instead of adenylate cyclase, it's going to activate phospholipase C. Once we have our activated phospholipase C, what that's going to do is it's going to split a plasma membrane phospholipid called PIP2 into two different second messengers. One of the second messengers is diacylglycerol, or DAG, and that's going to activate protein kinases just like we saw with cyclic AMP. The second messenger is going to be inositol triphosphate, or IP3, and what this does is it's going to cause calcium to be released from intracellular storage sites. Calcium release is really important because calcium ions themselves can act as another second messenger. So if we're talking about the PIP2 calcium, We have DAG that can go on and act as a second messenger, just like we saw with cyclic AMP. We also have IP3 that can act as a second messenger, and we also have calcium ions that can act as another second messenger. And calcium ions play huge roles, which you probably remember from skeletal muscle and your bone as well. So calcium ions can alter enzyme activity in channels, or they can bind regulatory protein called comedulin. Once you have calcium bound to comedulin, that can activate enzymes that cause an amplification response, just like we saw with our cyclic AMP cascade. So we do have other signaling mechanisms. These aren't as prominent as the PIP2 calcium and cyclic AMP, but we also do have cyclic GMP, and it is the second messenger for selected hormones. We also have other hormones that work without some sort of second messenger system. And one example is the insulin receptor. The insulin receptor is a tyrosine kinase enzyme, so it can phosphorylate itself. Because insulin's coming in and binding to an enzyme that's capable of phosphorylation, it doesn't need a second messenger. Its own tyrosines, it phosphorylates its own tyrosines when insulin comes in and binds. So the activated tyrosine kinases provide docking sites for relay proteins, and these relay proteins are responsible for initiating that cascade of phosphorylation that's going to ultimately trigger cellular responses. Okay, intracellular receptors, because remember, if we're talking about steroid or lipid-soluble hormones, we're talking about intracellular receptors. And intracellular receptors, along with the thyroid hormone, because that was our exception. What happens is that these receptors are typically in the nucleus in some sort of region of DNA. So the lipid-soluble steroid hormones and thyroid hormones bind to and activate intracellular receptors, again, because they're able to diffuse across the plasma membrane. Once they form the receptor hormone complex, once it goes inside of the cell and binds to its receptor, it's then going to enter into the nucleus and bind to a very specific region of DNA. Once it's bound to this region, it's going to help initiate DNA transcription to produce mRNA. And then mRNA can be translated into a specific protein, and these proteins synthesized have various functions, like metabolic activities, structural purposes, or could even be exported from the cell. And again, our one exception is the thyroid hormone when we're talking about amino acid base, but it's also an exception when we're talking about intracellular receptors, because the thyroid hormone receptors are actually bound to DNA. So they actually go through the nucleus themselves and bind to the receptor there. They don't bind to the receptor outside of the nucleus and complex before they enter the nucleus. So here's an image of everything I kind of told you in words. Here's our steroid hormone. It's capable of diffusing across the plasma membrane, which it does. So it comes inside. It binds to its receptor somewhere in the cytoplasm over here. Once we have the hormone bound to the receptor, it's able to enter the nucleus through the nuclear pores and bind to a very specific region of DNA to enhance transcription and translation of proteins. And these proteins that it produces have various functions. Like I mentioned before, there is one exception, that's the thyroid hormone, whose receptors in the nucleus are already bound to DNA. So the steroid hormone actually diffuses into the cytoplasm, and the hormone by itself diffuses through the nuclear pores and binds this receptor that's already attached to the DNA. The effect is still the same, to enhance transcription and translation and to produce proteins.